Recall that nervous tissue is dominated by two cell types. The first cell type was the neurons, which are the structural units of the nervous system, and they also fulfill the role of being the signal-sending cells of the nervous system. The second cell type is a cell type called the neuroglia, which are also called the glial cells. And these are the supporting cells of the nervous system because they help to maintain neurons in various ways. And this was because neurons were very highly specialized for sending signals, and they were very poor at carrying out other functions. And so it's the neuroglia that are the support cells for the nervous system. So here in this segment, we'll examine the neuroglia. So the neuroglia, which are also referred to as the glial cells, are non-excitable cells, which means that a stimulus will not change their membrane potential, and the glial cells will not release neurotransmitters. So this was in contrast to the neurons, which were excitable, and they would release neurotransmitters in order to stimulate and signal nearby cells that they were communicating with. So we see that the glial cells are not able to send signals like the neurons are. So instead, the neuroglia support and maintain the neurons in various ways. So first we'll examine the glial cells of the central nervous system, and then we'll take a look at the glial cells of the peripheral nervous system. So we see that there are four types of neuroglia that are associated with the central nervous system, so that is the brain and the spinal cord. These four are the oligodendrocytes, the astrocytes, the microglial cells, and then finally the ependymal cells. So the oligodendrocytes are the cells that wrap the axons of nearby neurons, which creates a covering over the axon that is referred to as myelin. And the function of myelin is to increase action potential speed. So neurons that possess myelin around their axons are able to transmit and send action potentials at a much more rapid speed down the axon than neurons that do not have myelin around their axons. The next cell type are the astrocytes, and these are named because they are star-shaped cells. And we see that they have a number of branch-like processes, making them kind of resemble stars. These branches reach out and cling to nearby neurons, as well as reach out and cling to nearby capillaries. And one of the functions of astrocytes is to help facilitate the exchange of nutrients between the small capillaries, the small blood vessels in the central nervous system, and the neurons that are nearby. The astrocytes also function to clean up excess neurotransmitters as well as excess potassium ions in the space that is surrounding the neurons. Astrocytes also control the formation of tight junctions in the blood vessels of the brain, and the presence of tight junctions makes the capillaries of the brain less permeable than typical capillaries, which means that only things that are lipid-soluble can move from the blood across the plasma membrane of the capillary cells to the brain tissue. These tight junctions that secure the capillaries inside the brain help to form or create what is called the blood-brain barrier. And this helps to prevent changes in blood chemistry from affecting the brain. At the same time, it also helps to limit which substances are able to leave the bloodstream and enter the brain tissue. Next, we have the microglial cells, and the microglial cells function in a capacity that's very similar to cells of the immune system. They function to break down dead or dying neurons in the central nervous system, and they also function to kill microorganisms like bacteria that have entered the central nervous system. Next, we have the ependymal cells, and these are cells that line cavities within the brain that are called ventricles. The ventricles are filled with a fluid that's called cerebrospinal fluid, and this is a liquid that acts to support and cushion the very soft and delicate nervous system tissue from within the brain, and even within the spinal cord as well. The ependymal cells have cilia on their surface, and the cilia are these small hair-like projections that wave and beat, and this helps to circulate and move the cerebrospinal fluid within the ventricles. So now that we've examined the neuroglia of the central nervous system, we can turn our attention briefly to the neuroglia of the peripheral nervous system. We see that there are two cell types here. We have the satellite cells, and we also have the Schwann cells. The satellite cells surround neuron cell bodies in the peripheral nervous system and function very similarly to astrocytes in that they help to exchange nutrients and things like that between capillaries and the neurons that they're associated with. Next we have the Schwann cells, and the Schwann cells wrap nearby neuron axons and function as myelin in the peripheral nervous system. So just like myelin in the central nervous system, the Schwann cells that form the myelin in the peripheral nervous system function to increase action potential speed in the axons that they are associated with.